Welcome to BioBits and Bytes. We're tackling a major environmental villain today, one that sounds like it should be locked away in a sci-fi movie archive. Forever chemicals.
It's a compelling and, frankly, terrifying nickname.
Yeah.
We are talking about polyfluoroalkyl substances, or PFAS.
Right.
These are a massive group of synthetic organic compounds, SOCs, that are defined by their incredible, almost impossible durability.
That durability is the hook. If you're preparing for a meeting, catching up on environmental science, or just curious, think of PFAS as the chemical foundation of materials like Teflon.
Gore-Tex, specialized firefighting foam.
Exactly. They were designed to resist everything, heat, oil, and water, and they do their job flawlessly, maybe a little too flawlessly.
That near indestructibility is precisely why they are now global contaminants. I mean, once released into the environment, they virtually never break down naturally.
Our mission today is focused entirely on the solution side of this problem. We are diving deep into the most promising, cost-effective weapon against this ubiquitous pollution, bioremediation. We're going to explore cutting-edge microbial solutions, understand the chemical limits of current biological treatment, and meet a specific, high-potential supermicrobe called Rhodopseudomonas palustris.
It's a really urgent conversation because, as we'll get into, the traditional methods for cleaning up PFAS are, well, they're expensive and deeply inefficient. We need a biological shortcut.
Okay, let's unpack the core problem using a quick analogy check.
Yeah.
The structural nightmare here is the carbon-fluorine bond, or CF bond.
Yes.
It is frequently cited as the strongest single bond in all of organic chemistry. I've always pictured it like a molecular vault holding the toxic contaminant prisoner. You need immense energy to crack that safe. Is that a fair way to think about it?
It's the perfect analogy. The CF bond is characterized by two things: a very short bond length and a high degree of polarity, meaning the fluorine atom pulls the shared electrons tightly toward itself. This makes the bond highly stable and resistant to chemical attack. It demands a huge amount of activation energy to break.
Far more than other bonds.
Oh, far more than, say, a CH or even a CCl bond. This inherent stability is the root of the forever problem.
And what happens when these incredibly stable chemicals enter our environment and subsequently our bodies?
Well, PFAS are the synthetic organic compounds we mentioned, and they are now linked to a wide range of adverse health effects in mammals.
Yes.
Liver damage, immune system disorders, endocrine disruption. And the sheer volume of this problem is staggering. They are ubiquitous. We find them in drinking water, groundwater, rivers, wastewater, household dust, and, most critically for today's discussion, heavily contaminated soils.
We need to understand their physical properties too, because that explains why they don't just, wash away.
Exactly. They are highly stable, as we noted, but they also possess a unique, somewhat confusing dual nature. They are amphiphilic.
Okay.
That means one end of the molecule loves water. It's hydrophilic, usually the carboxyl head. And the other end is both hydrophobic, so water repelling, and lipophobic.
Fat repellent.
Yes, fat repelling.
Wait, if something repels both water and fat, where does it go in a living system that is made up of water and fat? That seems counterintuitive.
That's a great question, and that's where it gets really interesting. Because they repel fat, they don't accumulate in fat cells like many other persistent organic pollutants do.
No.
Instead, their amphiphilic nature drives them to accumulate at interfaces. Specifically, they love binding to the surfaces of proteins and membranes within living organisms.
So they're not dissolving in fat, they're sticking to the machinery of the cell.
Precisely. They essentially attach themselves to the structural components of cells, leading to their persistence and bioaccumulation in places like the liver and blood serum.
So we have a global contaminant that is everywhere, designed to resist nature and structurally suited to stick around inside our biology. That defines the urgency of the problem.
It does?
Which brings us to the inadequacy of our current cleanup strategies.
Yeah.
Let's move to section 1, the urgent problem and the limitations of traditional cleanup. Where are we seeing the most severe contamination, and how has the world tried to clean it up so far?
The scale is immense, and the sources are diverse. You have industrial waste discharge, landfills, and infamously sites where aqueous film-forming foam, or AFFF, was used.
The firefighting foam.
The firefighting foam, exactly. It was the gold standard for fighting high-intensity petroleum fires. Think military bases, airports, refinery training grounds.
Right.
For decades, these sites were saturated, leaving enormous plumes of PFAS contamination in the soil and groundwater.
When contamination like that is identified, what does the cleanup response look like traditionally?
Historically, we rely on physical or chemical processes. The physical methods include adsorption, primarily using granular activated carbon or specialized ion exchange resins,
Then filtration.
and filtration, often a reverse osmosis or nanofiltration membranes. Then, on the more intensive end, we have thermal treatment, incineration, chemical oxidation reduction using strong reagents, and simple soil washing.
Okay, if these methods exist, why are they considered failures in the context of large-scale PFAS remediation?
They fail on three key fronts: cost, practicality, and, this is a big one, the creation of secondary waste.
Let's take adsorption.
Okay. Activated carbon is effective, but eventually the carbon is saturated with the pollutant.
And if adsorption is the solution, don't we still have the core problem? I mean, where does that filter full of forever chemicals go next?
That's it exactly. You've just shifted the problem from the water or soil to a spent filter media. That material then requires expensive, high-temperature thermal destruction,
Incineration.
or it becomes a concentrated toxic waste that must be safely landfilled forever. Thermal treatment itself is incredibly energy intensive, requiring 1,000 degrees Celsius or higher, and there are concerns about incomplete destruction leading to atmospheric release.
So high heat, high pressure, enormous logistical cost, and you're often left with concentrated waste. What about just, basic soil washing?
Soil washing is an ex situ method.
Meaning you have to dig it all up?
You have to dig up massive volumes of contaminated soil, transport it, wash it with solvents or water, and then deal with the resulting contaminated wash water. It's low-tech, but highly expensive due to excavation and transport costs.
And I imagine there's a huge risk of causing even more water contamination during the washing process itself.
A significant risk, yes, if the protocols aren't followed perfectly.
And the really critical gap in the status quo, which the sources highlight repeatedly, is that current commercial methods are overwhelmingly focused on cleaning up contaminated groundwater.
Yes.
We lack robust, affordable, large-scale solutions for contaminated soils.
That's the critical infrastructure gap. We have technologies that can address water, but soils are much harder to treat in situ in place. You can't easily pump a chemical oxidant through clay soil.
And you definitely can't incinerate 1 million tons of dirt.
Not without massive cost and energy expenditure, no.
That is precisely where the bioremediation promise steps in.
Bioremediation, the use of biological agents, microbes, fungi, plants to break down contaminants, is the environmentally safe, cost-effective alternative. It offers the potential for large-scale in situ cleanup of contaminated soils without the high energy requirements or the secondary toxic waste stream.
It makes perfect sense economically. If you can treat the contamination where it sits, you eliminate the massive post-cleanup costs associated with excavation, shipping, and specialized thermal destruction.
And we have a long, successful history of this technology. Bioremediation is the commercial standard for cleaning up other persistent contaminants.
Like oil spills.
Notably, petroleum hydrocarbons, yes. Also, chlorinated substances like TCE and PCE and various pesticides. The question is whether we can leverage that established biological prowess against the molecular vault of the CF bond.
And if we connect this to the bigger picture, the urgency isn't just about finding a solution, but finding a sustainable green chemistry solution.
Right. We need to avoid massive fossil fuel inputs for thermal destruction and embrace simple, environmentally safe processes that can operate in the natural environment.
That brings us to Section 2, decoding the microbial attack. If the CF bond is the strongest bond in organic chemistry, how on earth can a tiny, soft microbe muster the energy to break it open?
That's the central scientific challenge. Overcoming the massive energy barrier required for CF bond cleavage, a process called defluorination.
Right.
A microbe needs to find a way to catalyze that reaction with minimal energy input, or find an enzyme that can channel a huge amount of localized energy right into that bond.
We see two main theoretical strategies for microbes to achieve this: oxidation versus reduction.
Correct. Oxidation involves adding oxygen across the FC bond, often using an enzyme that incorporates atmospheric oxygen. This is a common strategy in aerobic bacteria.
And reduction.
Reduction involves adding electrons across the FC bond. This often takes place in anaerobic environments, powered by electron donors like hydrogen or organic matter.
Is the evidence for success still largely theoretical? Or are we seeing concrete microbial wins?
Well, while confirmed reports of complete end-to-end biological defluorination of long-chain PFAS are still limited,
Okay.
the evidence that the concept is viable is growing rapidly, particularly when we're dealing with precursors or smaller fluorinated molecules.
Let's discuss the early wins. What evidence proves that microbes can even interact with these compounds?
First, we have evidence of bioaccumulation. Certain bacteria, particularly Pseudomonas species, have been shown to simply suck PFAS into their cells,
They track them.
essentially trapping them in the cell biomass. This occurs under both aerobic and anaerobic conditions. Short-term experiments demonstrated that various Pseudomonas strains could remove 28 to 32% of PFAS compounds over just a few days.
Suggesting a strong adsorption mechanism.
A very strong adsorption mechanism, even if full degradation wasn't achieved in those tests.
That's a good trap. But what about the actual molecular scissors, the enzymes that cut the CF bond?
We have confirmed defluorination of monofluorinated compounds. This is the smoking gun proving the enzyme activity exists.
Okay, so a simpler molecule.
A much simpler molecule. For instance, Pseudomonas species can utilize fluoroacetate, a simple monofluorinated compound, as their sole carbon source under aerobic conditions. The defluorination process here involves an enzyme that replaces the fluorine atom with a hydroxyl group derived from water.
Let's pause and simplify that part again. We're taking FCH, CO here, and adding a hydroxyl ion, an O air,
Right.
resulting in HOCA plus a free fluoride ion. What does this one simple reaction tell us about the future of PFAS cleanup?
It tells us that biological CF cleavage is mechanistically possible. Nature has evolved enzymes, dehalogenases, that can perform this task. Our challenge now is moving from a simple one-carbon fluorinated compound to a long, complicated chain of 8 carbons, like PFOA, which has fluorine atoms all along its backbone.
We need more powerful, less specific enzymes.
Exactly, more promiscuous enzymes.
To understand the reduction strategy better, we often draw parallels to other halogenated contaminants, right?
Absolutely. Think about chlorinated solvents. We know how to break CCl bonds effectively using anaerobic pathways, a process called organohalide respiration, or OHR. In OHR, certain anaerobic bacteria use halogenated organic compounds as their terminal electron acceptors.
Like using an alternative battery source.
Precisely. They're basically breathing them in a process that breaks the halogen-carbon bond to gain energy.
And we have success stories here, too.
We do, moving beyond simple accumulation. For example, the anaerobic bacterium, Acidum microbium sp strain A6, has shown significant success using an electron donor.
What kind of success?
Over 100 days, this specific bacterium achieved a substantial 60% reduction in PFAS compounds by using hydrogen as the electron donor. This success highlights that supplying sufficient reductive power is essential for defluorination. The pathway is difficult, but the energy input is the key.
Now, here's where it gets really interesting, because the greatest breakthrough demonstrates that the CF bond can be broken even without atmospheric oxygen,
Yes.
which bypasses a major logistical hurdle for in situ cleanup.
This is the truly game-changing finding involving the denitrifying bacterium Thaur aromatica. Researchers identified a mode of oxygen-independent defluorination that completely degraded certain para-substituted fluoromatics.
An aerobic microbe that typically needs oxygen, but it breaks the CF bond without it. That seems like a massive paradox.
What's fascinating here is the sheer brute force the microbe employs. The organism utilizes a specific enzyme complex known as class I benzoyl coenzyme A reductase, or BCR.
Okay.
This complex is used for aromatic compound metabolism, but in this case it catalyzes the ATP-dependent defluorination of a fluorinated intermediate, 4-FBZ-CoA.
So instead of using ambient oxygen as a fuel source, the cell is using its own energy currency, ATP, to manually force the bond open.
That is the crucial insight. ATP hydrolysis, the breakdown of ATP to release energy, provides the immense activation energy needed to break that stubborn CF bond in an environment devoid of oxygen.
Wow.
The pathway also involves other enzymes like DCH and OAH. The final result of the degradation of 2F-benzoate is the production of HF, hydrogen fluoride, and CO2. The key step is an unstable intermediate that spontaneously decomposes, expelling the fluoride as HF.
Let's pause and really break down the significance of this Thaur aromatica finding for cleanup efforts.
The finding is monumental for environmental engineering because of the cost implications. For most contaminated sites, especially those deep in saturated soils or groundwater, providing and controlling oxygen levels is logistically complex.
And incredibly expensive.
Incredibly expensive. It requires massive aeration infrastructure. If we can harness an enzyme pathway that uses internal cellular energy, ATP, in an anaerobic or oxygen-limited environment to break the CF bond, we drastically lower the technical demands and cost of large-scale remediation.
You just need to make sure the microbes have enough food.
Enough organic material or electron donors to keep producing ATP.
It proves that the biological systems have the necessary machinery, We just need to find the right metabolic engine to run it.
Which leads us directly to our star candidate for that engine.
That's a great question. Let's break it down. Section 3, the metabolically versatile microbe. Rhodopseudomonas palistris. This organism sounds less like a bacterium and more like the ultimate Swiss Army knife for environmental cleanup.
It truly earns that nickname. Rhodopseudomonas palistris, or R. palistris, is a common, non-pathogenic soil bacterium, but its metabolic capacity is astounding.
How so?
It can switch between all four major modes of cellular existence depending on its environment. You have photoheterotrophy, photoautartrophy, chemoheterotrophy, and chematotrophy.
So it can survive and thrive in virtually any environment, with light or without, with oxygen or without, using organic contaminants or just using CO2.
Exactly. On top of that, it's beneficial. It fixes atmospheric nitrogen, and it naturally produces useful chemicals like precursors for bioplastics and even clean fuels like hydrogen.
Which makes it an ideal candidate for synthetic biology.
A perfect candidate. Not just a cleaner, but a potential resource generator.
And researchers weren't initially looking at R. palistris for PFAS, were they? They were looking at another incredibly complex, recalcitrant material, lignin.
Yes. That work led to the key insights. Lignin is the second most abundant biopolymer globally, and it's what gives plants their structural rigidity. Like PFAS, it's notoriously difficult to break down chemically.
And the research into how R. palistris digests lignin provided a massive clue about its potential to handle other tough compounds like PFOA.
A huge clue, yes.
Let's detail that lignin degradation pathway because that is the foundation for the PFAS potential.
A comprehensive multi-omics study looking at genomics, transcriptomics, and proteomics was done to understand how R. palistris consumes lignin breakdown products, or LBPs.
Okay.
Lignin units are generally grouped into H, G, and S types, differentiated by the number of methoxy groups.
What the researchers found was a metabolic superpathway.
Precisely. R. palistris consumes these three structural types of LBPs indiscriminately. It doesn't need three separate pathways. It funnels them all toward the TCA cycle, the cell's primary power generation hub.
And how does it manage this universality?
It relies on a set of what scientists call promiscuous enzymes.
Okay, let me see if I've got this. Promiscuous enzymes. That just means they're not picky. They'll grab onto a few different types of molecules.
That's a great way to put it. They're not specialized. They are flexible enough to accept and process multiple, structurally similar but not identical, substrates. These enzymes are derived from the coumarate pathway.
Can you give us an example of how that works in practice?
The study identified key enzymes like alcohol dehydrogenase, RPA-PU1205, and aldehyde dehydrogenase, RPA-PO4206. Their functional role is to systematically strip off specific chemical groups, preparing the complex lignin fragments for entry into the TCA cycle.
They're the versatile processing unit.
Exactly. They can handle slightly different inputs and still achieve the same output. Energy precursors.
And just like with the Thauer finding, there was another oxygen-related surprise in the lignin work.
There was. The research noted that R. palistris can perform vinylate serengate demethylation, removing a tough methoxy group even under anaerobic conditions. This is remarkable because, biochemically, demethylation is a reaction that typically requires dioxygen as a reactant.
Once again, it finds a way to perform a highly energetic, typically aerobic reaction in a low-oxygen environment. This leads us to the central aha moment of the lignin study, which is the key to unlocking the PFAS potential, the redox bottleneck.
The study found that R. palistris's ability to break down lignin is not limited by the amount of catabolic enzymes it possesses. The breaking machinery is abundant.
So it's limited by something else.
It's limited by redox homeostasis.
That's a dense term. Let's dedicate some time to defining redox homeostasis simply for the listener.
Okay. Think of redox homeostasis as the cell's ability to maintain the balance of its internal rechargeable batteries, specifically NAD+ and NADH. NAD+ is the depleted state. It's ready to accept electrons. NADH is the charged state. It's full of electrons. When a cell breaks down organic compounds, it generates NADH, full batteries.
To keep the metabolic machinery running, it needs to regenerate the empty batteries, the NAD+ bare.
So it can accept the next batch of electrons. If this recycling process slows down, metabolism grinds to a halt.
So when R. palistris is aggressively consuming tough compounds like lignin fragments, it's constantly filling up those batteries. It needs an incredibly efficient place to dump those electrons, an electron sink, to quickly convert NADH back to NAD+ NAD.
That is precisely the bottleneck. The finding was that R. palestris grows to significantly higher concentrations under anaerobic conditions, not because it prefers darkness, but because photosynthesis is a far more efficient electron sink than regular aerobic respiration.
Wow.
Photosynthesis allows the bacterium to dump those electrons rapidly, keeping the NAD plus NADH battery balance perfectly maintained.
That's a game changer for engineering it cleaner. If we can solve the electron recycling problem, we can dramatically increase the rate at which it processes tough chemical structures like the CF bond.
Now, let's shift to the PFAS interaction breakthrough. Researchers tested this metabolically versatile microbe against perfluorooctanoic acid, or PFOA.
And the results?
The results showed that R. palestris can remove approximately 44% of PFOA from the environment over 20 days.
That sounds promising, almost half the concentration gone in under 3 weeks. But how is it achieving this removal? Is it cutting the CF bond?
The primary mechanism, as the initial study suggested, is currently adsorption. The microbe traps PFOA by drawing the amphiphilic molecules into its cell membrane.
Okay, here's where we challenge the assumption of success. If the microbe is just trapping it, what happens when that cell dies or is consumed by something else?
That's the major current limitation, and it leads to the engineering necessity. The research clearly indicated that PFOA is released back into the environment due to cell lysis cell breakdown.
So right now, R. palestris is an extremely effective biological trap, but it's not yet a consistent and complete degrader.
Not yet.
That means the key to future direction is applying the redox insight from the lignin study to the PFOA cleanup. How do we take that efficient electron recycling system and channel that massive reducing power toward actually breaking the CF bond?
We need to figure out how to modify the genetic machinery to overcome two hurdles. First, forcing the reductive defluorination pathway, like we saw in Acidomicrobium, using the cell's enhanced redox power.
And
Second, dealing with the inevitable toxic byproduct of that cleavage, the fluorine ions. If we can channel the electron sink toward bond cleavage efficiently, we essentially turn the microbe into a highly powered internal decomposition unit.
let's talk about toxicity. That brings us to section 4, the toxicity paradox. It turns out that not all forever chemicals are created equal.
And some of the replacements for the regulated chemicals are causing big hidden problems.
This section analyzes a crucial comparison study between two types of perfluorinated carboxylic acids, perfluorooctanoic acid, PFOA,
The famous long-chain regulated compound.
and perfluorohexanoic acid, PFHXA, a shorter chain compound often used as a direct replacement chemical and, critically, often unregulated.
Right.
Why are regulators typically more concerned with the longer chains like PFOA?
Generally, long-chain PFAS are more persistent and have longer half-lives in the human body. They're also typically more potent toxins. Regulators have focused their efforts there, leading industry to phase in short-chain replacements like PFHXA.
Based on the assumption that they're safer.
Based on the assumption that they're safer because they are more quickly eliminated from the body.
So what did this specific study reveal about the immediate impact of these two compounds on freshwater microbial communities?
Researchers incubated freshwater lake samples and exposed them to both compounds at concentrations ranging dramatically from environmentally relevant low doses up to extremely high concentrations.
And the high-level conclusion.
Both compounds, when applied at high concentrations, significantly altered the structure of the microbial community. This means the overall balance, who was thriving and who was dying, shifted dramatically.
But the community didn't collapse entirely, right?
That's the surprise. Neither compound significantly affected the overall diversity, the total number of different species present. The ecosystem didn't lose species, it just radically changed the relative distribution. The community adapted by shifting to more tolerant species.
Which one proved to be the more potent chemical aggressor?
PFOA showed more potent toxicity across the board. The critical finding for policy is that PFOA significantly changed community structure even at that lowest environmentally relevant concentration of 100 nanograms per liter.
Wow.
PFHXA, the replacement chemical, needed that extremely high 10 milligram per liter concentration to cause comparable structural changes. PFOA is clearly a more disruptive agent at real-world pollution levels.
What was the specific impact on growth? Did the community size shrink?
Yes, both compounds significantly inhibited microbial growth, measured either by total bacteria count or by overall biovolume. PFOA consistently reduced total bacteria counts 13 to 21% more effectively than PFHXA at comparable concentrations.
So more potent, more disruptive, more inhibitory.
That's right. It aligns with the long-chain hypothesis. But then came the respiration surprise, the activity measurement that defied the initial hypothesis.
This is the most counterintuitive result.
It is. The hypothesis was that a potent toxin would inhibit microbial activity measured as CO2 production or respiration. However, neither PFOA nor PFHXA showed significant inhibition of microbial respiration over the 4-week period.
That is baffling. If these compounds are toxic enough to kill some bacteria and significantly inhibit the growth of others, shouldn't the community's overall breathing rate slow down?
You would certainly expect that. But the results were even stranger. PFHXA at the highest concentration showed a significant acute stimulating effect during the first week.
A spike in CO2.
A spike in CO2 production.
What's the leading interpretation for that strange initial CO2 spike?
The interpretation speaks volumes about the toxicity mechanism. The proposed reason is that PFHXA is so acutely toxic to sensitive bacteria that it causes them to die quickly.
Okay.
As those sensitive cells die, decomposers, other microbes in the community, immediately begin breaking down the dead cells, releasing a burst of easily metabolized carbon, which registers as a spike in CO2 respiration.
So the stimulation isn't a sign of microbial health, it's a sign of a microbial graveyard.
Exactly. It's a toxin creating a feast for the survivors. The overall metabolic rate might have been inhibited, but that effect was masked or even outweighed by the surge of CO2 from the decomposition of dead biomass.
And the researchers also identified specific indicator species. Why is that important?
It gives environmental monitoring a new tool. If you sample a body of water and find elevated levels of specific microbial families, you can infer the type and concentration of contamination present.
Like a fingerprint.
Like a biological fingerprint, yes. They suggested that elevated cyprospiracy and pyrrolylacea may indicate high PFOA, while a high abundance of maraxylacea might indicate PMHXA.
What do you think about the policy implications here? PFHXA is currently unregulated because it's a short-chain compound, yet it clearly shows concerning effects.
I think the data strongly suggests a massive policy loophole. While PFOA is clearly more potent at environmentally relevant doses, the fact that PFHXA is rapidly becoming the industry replacement means its overall environmental concentrations are destined to increase dramatically.
Because it's unregulated, it's being released in greater volumes.
Exactly. We cannot focus solely on long-chain PFCAs. The potential for wide-scale disruption caused by the short-chain replacement chemicals justifies an urgent regulatory shift to classify all PFAS as contaminants of concern. Even mild chronic effects spread widely can be devastating.
This research makes it clear. Relying on a single, naturally occurring microbe to slowly eat up a complex PFAS molecule might take centuries. We need to combine forces.
Right.
That brings us to Section 5. Next steps in bioremediation and synthetic biology, accelerating the timeline.
Since single-shot methods are typically slow and incomplete, the cutting edge of remediation is focused on combined strategies, often called a treatment train, to maximize efficiency.
Let's start with the combination of natural biological systems.
A core combination is phytoremediation, combined with enhanced bacterial activity. Phytoremediation involves using plants, usually fast-growing, robust species, to naturally bioaccumulate PFAS.
So the plant acts as a physical pump concentrating the pollutant.
Exactly, sucking them out of the soil and storing them in their roots and leaves. Simultaneously, you introduce or enhance microbial degradation around the root zone, the rhizosphere.
Which is a perfect spot for microbes.
It's perfect. The roots exude natural organic carbon, which biostimulates bacterial activity. This is often done in a constructed wetland system, providing optimum conditions.
We also need to talk about microalgae. They're another promising biological agent in this treatment train.
Microalgae are incredibly versatile. They show promise for bioaccumulation, biodegradation, and bioadsorption of contaminants. Their key advantage lies in their photosynthetic function.
They supply oxygen.
They enhance aerobic bacterial activity by supplying two vital ingredients, oxygen and dissolved organic matter.
That's a crucial synergy. The algae breathe out oxygen, which powers the aerobic bacteria to attack the contaminants, and the bacteria in turn may release CO2 and nutrients that the algae need. It's a beautifully closed-loop, self-sustaining cleanup system.
Moving beyond natural synergy, we are seeing new materials designed specifically to enhance this biological approach. We have the novel Repimer platform.
Renewable artificial plant for in situ microbial environmental remediation.
That's the one.
That acronym sounds incredibly high-tech. But the source material is surprisingly low-tech, plant waste.
It is. It's based on chemically modified lignocellulose, derived from abundant and renewable sources like corn stover, the leftover plant material after harvest. This platform executes a 3-step cleanup cycle that solves the critical problem of secondary waste.
Walk us through the 3 steps of the Raffimer system.
Step 1 is adsorption. The lignocellulose is chemically modified to create a highly porous, large surface area framework that efficiently adsorbs the PFAS molecules. It traps the chemicals immediately.
Step 2, creating the perfect microbial home.
The Raffimer framework holds the PFAS captive right next to an ideal food source, simultaneously providing necessary nutrients for a specific microbial agent to grow: white rot fungus.
And white rot fungus is known for being a powerful decomposer.
It is. It has these powerful non-specific enzymes that break down recalcitrant compounds.
And step 3, the elegant final step, degradation and waste elimination.
The fungus then degrades the adsorbed PFAS. Experiments show removal rates exceeding 98% for PFOA and PFOS, and critically, the fungus degrades the lignocellulose framework itself,
Which eliminates the secondary waste problem.
completely. You deploy the system, the fungus eats the pollution and the delivery vehicle, and all you're left with is CO2 and harmless fungal biomass. It is a truly sustainable, closed-loop solution.
That level of synergy is required to tackle a problem this widespread.
Right.
But to get the fungus, or our friend R. palustris, to fully degrade these compounds, we need to know exactly how they're doing it.
Right.
And that requires advanced tools for discovery.
Absolutely. We need to map out the complete, currently unknown degradation pathways for PFAS. 2 advanced microbial ecology tools are guiding this research.
First, let's talk about stable isotope probing, or SIP.
Stable isotope probing is a detective tool. It uses stable isotopes like carbon-13 to feed the microbial community. If researchers use C13 labeled PFOA, for example, only the microbe that actively degrades that PFOA will incorporate the C13 carbon label into its cellular component.
Into its DNA or RNA.
Exactly. You can then extract that labeled DNA, sequence it, and definitively link a microbe's identity to the specific function of degrading the xenobiotic. SIP is how we pinpoint the individual hero microbes in a vast, crowded soil community.
And second, the omics approaches the whole systems view.
That's where we gain massive inference power: metagenomics, transcriptomics, and metabolomics. Metagenomics looks at the entire genetic potential of the community. Transcriptomics identifies which genes are actively being expressed when the microbe is exposed to PFAS.
It tells us what the microbe is trying to do.
Exactly. And metabolomics identifies the intermediate products being created, which helps build the step-by-step biochemical pathway map, a map that is currently missing for PFAS.
Once we have that map, we can return to the ultimate goal: applying synthetic biology to turn our versatile friend, Pseudomonas palustris, into a dedicated PFAS destroyer.
The initial lignin study already provided a crucial testbed for this. They successfully demonstrated the first application of CRISPR interference into R. palustris,
And CRISPR is a powerful tool to knock down or dial down the expression of specific genes.
without permanently deleting them, yes.
What did that initial use tell us about the microbe's engineering constraints?
It definitively confirmed the redox bottleneck hypothesis we discussed earlier. They selectively knocked down various genes associated with maintaining redox homeostasis, like PARH 650, proved essential for growth.
And the catabolic enzymes.
In sharp contrast, knocking down catabolic enzymes, like RPA41086 or RPA4696, showed little to no reduction in growth.
That is fundamental for the engineering roadmap. The cell already has enough degradation machinery. Trying to overexpress the catabolic enzymes won't work. We need to power the metabolic engine better.
Precisely. The future engineering goal for R. palustris is a sophisticated 3-pronged attack.
The first goal, improving the trap.
We must improve PFOA retention. We need to prevent the PFOA from being released upon cell lysis. This could involve engineering the cell membrane structure itself or genetically reinforcing the cell walls.
The second goal, boosting the internal power.
Genetically engineering the microbe to enhance CF bond degradation capabilities using that powerful redox insight. We need to channel the immense reducing power generated by the cell's metabolism more effectively toward the defluorination reactions.
And the third goal, dealing with the secondary toxin, fluoride ions.
Fluoride toxicity is a serious consequence of successful defluorination. Intracellular fluoride is toxic because it interferes with key metabolic processes.
So we have to get it out.
We have to get it out. We must genetically engineer the microbe to better handle the toxic byproduct. Researchers are focused on boosting the expression of Phe-afflux proteins, essentially giving the microbe a high-capacity pump to rapidly spit the toxic fluoride out of the cell as quickly as the PFOA is broken down.
That's the roadmap for scalable biological PFAS cleanup. Trap it better, eat it faster by powering the redox engine, and spit out the toxic byproduct faster. It demonstrates that synthetic biology is not just useful for discovery, but essential for application. That was a tremendous deep dive into the chemical constraints, the microbial heroes, the policy paradoxes, and the cutting-edge tools. Let's quickly recap the three main takeaways for the listener to internalize.
First, the PFAS challenge is fundamentally defined by the CF bond, which demands vast energy to break. This makes traditional cleanup methods costly, energy-intensive, and prone to creating secondary toxic waste. Bioremediation is the simple, environmentally safe alternative we urgently need.
Second, microbes offer genuine hope, proven by specific findings like the 60% anaerobic reduction by Microbiome and the discovery of oxygen-independent CF cleavage by Thaur aromatica. Our key engineering candidate, Rhodopseudomonas palistris, is an excellent trap, and its lignin catabolism shows that redox homeostasis is the key bottleneck we must solve.
And third, toxicity is complex. While PFOA is the more potent long-chain toxin, the short-chain, unregulated PFHXA is concerning. It causes significant structural shifts in microbial communities, severely inhibits growth, and its acute stimulation effect suggests rapid cell death. This demands an immediate policy shift to regulate all short-chain PFAS.
If we leave you with one provocative final thought to mull over, the most advanced microbe we have, our palistris, currently releases the PFOA back into the environment when its cells die. If we achieve the goal of engineering these cells to absorb the forever chemicals on a massive scale, in constructed wetlands or large soil remediation sites, we are creating billions of tiny biological containment vessels. The future challenge isn't just if we can get them to eat the chemicals, but how we can manage the life cycle and the subsequent cell lysis of these engineered microbes in a massive real-world system to ensure the PFAS isn't simply re-released into the water or soil weeks later. The engineering must account for both the chemistry of life and the chemistry of death.
A critical step for the environmental engineers.
Thank you for joining again.